Hi, everyone, and welcome. This is our first lightning talk round. In this session, we'll feature 6 10-minute restoration-focused talks. Same as before, please add your questions to the Q&A box and upvote the questions you'd like to hear answered live. Questions we don't have time to get to will be added to Whova so that speakers have an opportunity to address them. So first up today, we have Liz Koziol, who is an assistant research professor at the University of Kansas. And Liz will be talking about long-term effects of native mycorrhizal fungal addition in tallgrass prairie restoration. So Liz, please go ahead and share your screen.
Thank you for that introduction. How does that look?
So I can see your presenter view.
Okay, let me swap those. How does that look?
Perfect.
Great. Well, thanks for coming today, everyone. I primarily work in the tallgrass prairie, and my goal for today is to convince you that including not only seeds from these systems, but also microbes from these systems is the key to restoration success. And so I'm a mycologist. I study mycorrhizal fungi, and I'd like to give a brief introduction into the predominant fungi in grasslands, which are called arbuscular mycorrhizal fungi. And these fungi don't make mushrooms. They are subterranean, meaning they spend their entire life cycle below ground, but they're extremely important. They associate with about 85% of terrestrial plants and most prairie species. And so these fungi come in two forms. On the top, the picture of the beautiful yellow and white circles, those are the spores of the fungi, which are kind of like their seed. And so I spend a lot of time with the microscope sorting these by different color, shape, and size, which is how we tell the species apart. And the main function of these fungi lies in their other form, which is hyphae. And so in the bottom photograph, that white filamentous structure, sort of like a web or a netting or almost like a road network, is the hyphae. And so it's a long filamentous tube, which the fungi use to collect soil nutrients and then ultimately pass it to a plant host. And so the plant and AMF symbiosis begins when a plant seed starts to germinate and its little rootlets grow into the soil and interact with that fungal hyphae. The fungi actually grow inside of the plant root, and it's a mutualism, so both partners are getting something. The plant feed the fungi carbon, which is required for the fungi to grow, and then the fungi use their vast hyphal network to collect nutrients from the soil and feed it to their plants. And so plants can actually grow larger in associating with fungi due to that increased foraging area provided by the fungal network. And so I study fungi in these two environments, on the left representing the tallgrass prairie, and on the right representing our pre-restoration landscape, which often occurs after massive disturbance of the soil in the system. So this is a totally disturbed system representing a row crop agricultural field. And in many types of soil disturbances, what we see is they are really harmful for mycorrhizal fungi. For example, tillage can chop up those hyphal networks and kill those fungi. Planting and crop monocultures and chemical applications can also render them less abundant and less diverse. And as I will show, these changes in mycorrhizal communities can mean life or death for different plant species. And we can test this in the greenhouse by doing a mycorrhizal response assay, where we grow a plant with and without its native AM fungi. And so if you have a low mycorrhizal responsiveness, you don't really care about your fungi. And if you have a mycorrhizal response of, let's say, 15, you grow 15 times larger with your fungi than without. And so if we test this across a successional gradient, where on the left are very early successional plants, like native sunflower, weedy things, and non-native plants. And on the right are more conservative plant species that are typically only found in our remnant prairies. Here's what we find. So every dot on this graph is a different plant species organized by their successional stage. And generally, weeds and non-native plants don't care about their native prairie fungi, whereas these late successional plants are really strongly dependent on their native fungi. And these plants are often the ones that we see not being recruited from our seed mixtures. And so let's take a few plants from this data set with a range of mycorrhizal responsivenesses and imagine how this microbial filter can limit seed establishment and restoration. So if we take on the far end of early successional Rudbeckia hirta, the black-eyed Susan has a mycorrhizal responsiveness of about 1, doesn't really care about fungi, versus in my range, rattlesnake master has a mycorrhizal responsiveness of about 10. So if we take, for example, all these seeds and throw them into our restoration environment, which has a disturbed mycorrhizal community, what we would expect to see is a ton of these early successional plants that aren't reliant on mycorrhizae to establish really well, and fewer of those later successional plants to establish. However, in our restoration, if we transplant not only seeds, but the microbes that those plants are dependent on, what we'd expect to see is more of these late successional plants being able to establish in restoration. And so we call this movement of inocula of microbes, we call that a microbial transplant, and the stuff we're moving is inocula. And so we've done this a ton of times in the tallgrass prairie and beyond in different restoration projects throughout the US. And I don't have time to discuss all of the amazing results we found so far, but I'm going to highlight a couple case studies to show you some examples of how these fungi work. And so the first case study is a set of restoration experiments we did in 5 states in the US, from Oklahoma to Illinois. And at these sites, we inoculated with 3 different microbial treatments. So we included no microbes, which represent conventional restoration practices. We included whole prairie soil inocula, which is collected from a remnant prairie, an old growth prairie, and has everything in it from mycorrhizae to rhizobia. You can see in this photograph, even some biota such as worms. We also isolated only the AM fungi from that prairie soil. And so we had these 3 different inocula, and we applied them on the roots of our nurse plants. So we used little seedlings here. We grew in tiny pots with a background soil, and we inoculated about 10% by volume with our microbial treatments, either just AM fungi, whole soil, or none. We also seeded the whole sites with a 60 species seed mixture. And so here are our findings the first year. Let's look at the seedlings. On the top is survival, and on the bottom is leaf production or plant growth. And so in the black with our AM fungi alone versus our whole soil in gray, we saw improved survival and growth with microbial addition at the site. We shifted to what happened to the recruited seed community. This is some data from the end of the 3rd year. On the top is plant diversity, and on the bottom is non-native plant reestablishment. And what we saw was that plots that had AM fungi alone had increased diversity and reduced colonization by the local non-native plant at a given site. And so this data is amazing. It shows that native fungi can be a useful restoration tool. And it also shows that native fungi alone work almost as well, maybe even better than in some cases, than whole prairie soil, which is meaningful because our remnant prairies are rare and we don't want to dig them all up to be used as inocula. So now that we know mycorrhizae are a really important microbe to add in our restoration, the next thing we wanted to ask was, well, how much do you need to add? And I like to refer to this case study as the handfuls or dump trucks study. So we had a restoration site that was an old brome field. We added a seed mixture to the entire site, and then we installed this inoculation density gradient where we had plots with no fungi representing conventional restoration practices, low densities of fungi all the way to essentially dump trucks per acre. And so I'm going to jump ahead to what happened at this site year 7. These sites are really beautiful, these restoration plots. There's a ton of forb and legume diversity. That's one of my favorite plots to go sample at. But here's some data. First, I'll show you my graphs. I'll walk you through my graphs a little bit. On the Y here is going to be whatever thing I measured. And on the X is my inoculation gradient. So here, no fungi are in white. And then as you go this way, it represents adding more and more of that native mycorrhizal fungi to the plots. So these are just 4 different ways of looking at the exact same thing, which is plant diversity. FQI, total CC, Shannon, and Simpson all show the same thing, that the more native fungi you add, the more diverse your plot is going to be. If we dove a little deeper into what plants we're establishing, the non-native plants on the left versus the native plants on the right, we found that non-native abundance and richness declined, whereas late successional plant abundance and richness increased with increasing densities of land fungi. And when we dug even deeper into that data, we found that this was driven by late successional legume establishment in these pots. So after today, I hope I've convinced you that the key to successful restoration of these systems is to think beyond seeds and to consider microbes in a restoration. And there's evidence that this technique works in other systems across the world. So with that, feel free to reach out to me at michaelbloom or michaelbloom@gmail.com. Thank you.
Thank you so much for that awesome presentation, Liz. I don't think we have time for questions, but there are quite a few questions that we will paste into Whova if you're willing to go answer them. So next up today, we have Amy Gill, and they are a PhD candidate at Colorado State University, and they'll be talking about seed mix design and planting techniques to improve pollinator abundance in conservation reserve program plantings in Colorado. So please go ahead and share your screen, Amy.
You're muted, Amy.
Okay, yes, I figured. Do you guys see my main screen or in presenter view?
You're good. We see your main screen.
Okay. Let's see, but I cannot see you guys now. All right, anyways, I'll start. Hi guys, my name is Amy Gill. I am a fourth year PhD student, and today I'll be talking about seed mix designs and planting techniques in conservation reserve program plantings across Colorado. And I'd like to especially thank all of my collaborators, especially my advisor, Dr. Kerry Havilla, and all of the team members across CSU and USDA ARS, and Dr. Lauren Perensky and her team members. So I don't know if the screen is moving because it's not moving on my end and I don't see you guys.
We still see your title slide.
Okay, let me pause share for a second. How about now? Okay, there you go. Is that working?
I still see your title slide.
Oh my goodness. Okay, sorry. Oh no, that's so bad.
You're good. Would you like me to share your presentation? I actually have a copy of it open.
Sure.
Okay.
Good. Thank you. Sorry about that.
No problem.
I don't know why it's just messing up with the screens. Yes. Okay, so if you want to move to the next slide. Yeah, so you might have heard, sorry about that, guys. I'll walk through as quickly as I can. So you might have heard that pollinators are dying and bees are disappearing. Especially in US, we have about 19% of annual loss of most of our iconic butterflies and 30% loss of what do you say, just like bee population. And what that means for our ecological benefits is that we are 1/3 of our agriculture food productivity is dependent on that. So is our wildlife habitat and animal pollination. And in general, human health is what do you say, dependent on bee pollination, and we are losing a lot of that because of modernization, climate change, and loss of habitat. Next slide. So this is where Conservation Reserve Program plantings come into play, where CP42 is an initiative through Conservation Reserve Program plantings, which is a cost share land-based restoration program across all of the US, but in particularly for this chat, this talk will be focusing on pollinator plantings and their role in standard pollinator plantings. I think you'll just have to scroll. There's an animation part to it. So, but they tend to fail across Colorado just because of post-ag agriculture and harsh growing environments. So we need innovative approaches such as diverse and alternate seed mixes and planting techniques. And this is what this research is on. And to do this, we set up a field experiment across 5 sites in Colorado, which was over 3 counties: Weld, Washington, and Latimer County. So, and we asked 2 main questions: whether our restoration treatments improve flower number, and what does that mean for bee visitation. Next slide. So we deployed a total of 4 treatments. The first one was business as usual or standard, which was counted specific seeding mixes and practices. For our second treatment, we had manure slide in addition to our standard BAU. And for the third one, we had a targeted weed control by planting our business as usual treatment in different strips of grasses and forbs. And our fourth treatment was alternate seed mix, which was just a drought tolerant, diverse, forb heavy seed mix. And we collected data on flower abundance, flower open inflorescence, pollinator diversity, and abundance, in addition to a lot of other data collection. Next slide. I would like to move to the next slide. Okay, so let's talk about the first result, and next slide. So here on what we see is that on the x-axis we have all of our treatments, where business as usual is gray, yellow is manure, strip planting is in black, and alternate treatment is purple. On the y-axis we have number of flowers, and on the top we have early, mid, and late season. Seasonality is important because of all of the environmental factors and how it affected the treatments across different time of the years. And to measure a success rate was to declare at least 3 flowering plants across all of the seasons. So we had early, mid, and late season. What we see here is on the top, in early season, we see that all of our non-standard treatments, so the bar in yellow and the bar in black is manure and strip planting. They increase flower abundance in early season and in mid-season when compared to our control treatment of business as usual, which was light gray here. Whereas in early season, we see that our alternate treatment did actually worse than all of the other treatments across all of the seasons. Whereas later into the season, it improves, even if it does not significantly improve the number of flowers. So what we find here is actually that earlier into the season of early and mid season, our abiotic treatment, where we improve soil integrity by adding manure, and we do a targeted weed control that actually improves the floral resources that could be utilized for potentially be used for pollinators. Whereas later into the season, seasonality becomes more important because it's a harsh growing condition, and that's when we did not find any significant difference of treatments on number of flowers. Next slide. So this is just an example of one of our sites. On the right, we have a picture from 2018, and on the left, we have a picture from the very next year in 2019 from a similar spot, similar site, and we can see a bunch of kochia that has taken over. So really it comes down to initially, just like as dryland restoration depends on seasonality and what the site conditions look like. So yeah, so success is really determined by, at least initially, by the growing conditions of the site. Next slide. Yeah, for our second question, next slide, we'll be talking about bee visitation. And what we find here is, well, it's missing the y-axis here, but on the x-axis, again, we have the treatment. On the y-axis, we have number of bee pollinators. And what we find here is, on this graph, is just late season. And we see that all of our non-standard treatments, so manure treatment, strip planting, and all treatment, increased bee visitation later into the season, whereas early and mid-season did not have a significant difference of our treatments. But what we see is that maybe later into the season, once the site has stabilized and established enough floral resources, bee pollinators are attracted. For the sake of this talk, I'm only discussing bee visitation, but we also looked at non-bee pollinators and also diversity analysis for that. And next slide. So basically, I'm just like wrapping up that our manure and strip planting tend to increase early and mid-growing season flower abundance, and later into the season, non-bee standard bee visitation increases by non-standard treatments. Thank you, and sorry about technical problems.
No worries. Thank you, Amy, for that great talk. We don't have time for questions, but again, you have a few questions and we'll post them in Whova if you don't mind going to checking them out. So next up, we have Laura Shriver joining us from the USGS to talk about RestoreNet. And I'll be sharing my screen here. Just give me one sec.
Awesome, thanks. Good morning, everyone. My name is Laura Shriver. I'm an ecologist at the USGS in snowy Flagstaff, Arizona. And today I'm going to talk to you about lessons we've learned from the past 5 years of RestoreNet networked restoration experiments. Next, please. So let me start by introducing you to the program I coordinate, RAMPS. RAMPS stands for Restoration, Assessment, and Monitoring Program for the Southwest. And our mission is to strengthen restoration outcomes by providing science and guidance on effective restoration strategies. Next, please. The RAMPS program was created to develop and share strategies to restore the increasingly degraded ecosystems of the Southwest in the face of climate change, which can further exacerbate the challenges of aridity in this region. Next, please. So RAMPS has a 3-step process to improving restoration. We start with partner engagement, or the roots, to assess the needs of land managers and restoration practitioners in the Southwest. Next, based on those needs and ideas, oh, sorry, I was saying next for myself, not for you. So based on those needs and ideas, we conduct research aimed to produce actionable science and inform best management practices. Lastly, we share results with project partners and the wider restoration community through things like publications, research briefs, newsletters, and presentations. Now, next slide, please. So I mentioned our partners, and this graphic shows just some of them. Our collaborators are essential because they share research needs with us and partner with us to co-produce science. Next, please. So the main RAMPS project I'd like to talk to you about today is called RestoreNet. RestoreNet is a co-produced research network of experiments that systematically test restoration treatments across environmental gradients in the Southwest. Next, please. So what I mean by networked is that we have experimental sites throughout the Southwest that test the same treatments across environmental gradients. So this allows us to explore how treatments work across sites and how treatments interact with gradients like temperature and precipitation. We currently have 25 sites spread across 7 ecoregions in the Southwest and plan to add more in the next few years. Next, please. So RestoreNet benefits land managers in several ways. First, we co-produce knowledge as we assess land manager needs to decide which treatments to test, interpret results, and explore new ideas. The plots also serve as demonstration sites with examples of restoration techniques. Lastly, the sites are low risk, as small test plots can reduce cost and build understanding that later supports larger scale projects. Next, please. RestoreNet also improves restoration outcomes. Through testing the same treatments across a diverse network of sites that span environmental gradients, so each site has unique characteristics, disturbance histories, and land management needs. And by placing sites across gradients, we can see how environmental characteristics interact with restoration treatments. This way, we can produce best management practices for individual sites and identify strategies that work across ecoregions or the whole network. Next, please. So first, I'm going to talk to you about what we call RestoreNet 1.0, which was the round of treatments that lasted from RestoreNet's creation in 2018 through 2022. Next, please. So we did seeding trials with two types of seed mixes. We compared what we called a cool mix of species that are adapted to historic conditions of a site to a warm mix from hotter, drier areas that have the potential to do better under projected future hotter, drier climates. Next, please. We also explored soil surface modifications designed to improve seeding success. These included soil pits, which are small depressions in the soil surface that gather moisture, wood mulch, which can also increase moisture, and conmods, which are small fence-like structures that mimic nurse plants by inhibiting erosion and keeping seeds in place. Next, please. So getting into a brief summary of results, first, we found that the cool mix did best. However, this was during a wet year, so our current efforts involve seeding for current and future climates. Next, please. We also saw that soil surface modifications improve seeding success. Pits did the best of all three treatments, and both pits and mulch were shown to increase soil moisture, which probably led to more successful seedling establishment. Next, please. We also saw that precipitation mattered, with increased precipitation right after seeding and increased cumulative precipitation, both improving seeding outcomes. Next, please. And last, we saw that while exotic species didn't impact seedling emergence, they did negatively impact seedlings once they were established. Next, please. So now I'll briefly talk about our outplanting experiments where we grew seeds in the greenhouse, then outplanted them as seedlings into the field at a subset of RestoreNet sites that spanned an aridity gradient. And some of the greenhouse grown plants were also harvested for trait screening. Next, please. So getting into brief results, in the outplant experiments, we saw that certain plant traits were associated with higher outplant survival. Next, please. We also saw that trait variation was restricted at the harshest, most arid sites, indicating that the aridity at these sites may act like a filter that selects for only a few traits that can confer success in this environment. Next, please. So what this all means is that matching traits to an environment and the ecological conditions at a restoration site has the potential to improve outplant success. So for instance, in the Colorado Plateau, we found that plants with thick leaves and thin roots did best across all environments, and plants with traits that confer drought tolerance had the highest survival at the most arid sites. Next, please. So now I'll briefly touch on RestoreNet 2.0, which is our current round of experiments, which started in 2022. Next, please. So in 2.0, we're exploring a wide range of treatments aimed to increase the success of seeding and improve soil health. This diagram shows the 2.0 treatments and their intended benefits. So we're going to combine seeding treatments with the soil pits that we knew did well in RestoreNet 1.0 by increasing moisture, seed balls to increase contact with seed resources, site-specific live topsoil inoculation with soil collected from nearby reference sites to boost beneficial microbes, and at a subset of sites, we're going to delve into targeted livestock grazing to create microtopography and increase seed contact with soil resources and nutrients. Next, please. We can skip this one. We do outreach and science communication. So thank you for listening. I don't know if I'll have any time for questions, but my email is here, so you can reach out to me. And there's a QR code and link to the RAMP's website on the left, and a QR code to sign up for our newsletter on the right. Thank you.
Thank you, Laura. I do think we have time for a couple questions. So first up, was the manure raw or composted?
We didn't use any manure. We used wood mulch from pines as a treatment to increase soil moisture. And in our current iteration, we're using live topsoil inoculation, where we collect live soil from a reference site for each RestoreNet site, bulk it in the greenhouse, then apply it as a treatment in the field. Oh, sorry, you're talking about probably the poop from the cows. So we're not actually applying manure, but we're going to have the cattle in an enclosure for a certain amount of time, where we're hoping they will give us some manure. So it'll be raw straight from the cow.
Gotcha. Okay, can you, oh, this one is, can you go back to the target species at the end?
Species.
Back at the RestoreNet 1.0, probably.
Oh, yeah.
These?
Yeah, so this is an example seed mix for the Colorado Plateau. We had seed mixes that were specific to each region and had cool and warm species for each region in 1.0. So we had these species for the Colorado Plateau, but we also had unique mixes for the Sonoran, the Mojave, the Chihuahuan, the Madrigan Archipelago, and I think that's it.
Awesome. So last question, have you conducted any sagebrush research in a higher saline soil environment?
Yeah, that's an interesting question. We haven't seeded sagebrush as one of our species because most of our sites are kind of below the sagebrush line. We are working on an experiment to try to figure out restoration techniques in a saline sodic area at Wupaki National Monument right now, but that's not really an area sagebrush is growing either as much.
Thank you. There are some more questions that will be posted in Whova.
Awesome.
So now we need to move on to our next speaker. Sarah Kotsopulos from the Toronto and Region Conservation Authority will be talking with us about the Meadoway. And there we go.
Yeah. Okay, thank you, Gwen. So hello, everyone. My name is Sarah Kotsopulos, and I'm a technologist at the Toronto Region Conservation Authority, and I'm going to be introducing you to the Meadoway project. Next slide. So for reference, the Meadoway is located in Toronto, Ontario, up here in Canada. It encompasses 16 kilometers of hydroelectric corridor that runs through the city of Toronto, so it's a very urban restoration project. Next slide, please. The aim of the project is to take those barren turfgrass fields that you saw on the last slide and turn them into a combination of meadow habitat and a paved trail that runs the 16 kilometers from downtown Toronto to the Rouge National Urban Park in the east. The objectives are to increase biodiversity, increase people's exposure and connection to nature, and provide a corridor that connects urban green spaces and communities. This project was piloted starting in 2012, but officially began in 2018, with the last areas being seeded this year. I will be speaking about the restoration methods portion of this project. Next slide, please. So site preparation is one of the first things done on our sites, and it's one of the most important steps to any seed-based restoration project, especially for one in an urban center like ours with lots of invasive species present. We've used numerous strategies to prepare our sites, but have moved towards multiple rounds of mechanical tilling and seeding with a cover crop, as you can see here in these images. This process is repeated 4 to 6 times and is spread between 1 or 2 years, depending on permissions and timeframes. And this is done to try to deplete the seed bank of undesired species so that it's ready for native seeding. Next slide, please. So once the site prep is completed, the seed is typically drilled into the area in either the spring or the fall, and then adaptive management activities are started to help establish and maintain the native meadows. Next slide, please. So as I'm sure most of you know, our seed mix designs are influenced by multiple factors. Soil conditions is a pretty obvious one that changes what species will grow and be successful. Things like soil type and texture and degrees of wetness are all considered. Despite being what seems like a fairly uniform space, there's many wet pockets, dry sandy slopes, and areas with a lot of gravel. So designing seed mixes that are specific to the conditions they're going to be sown into greatly increases those germination rates. Invasive species presence can also impact what species we use because we may have to use a mix that contains very aggressive growing native plants so that they can withstand the competition. Although we try to control for invasives during our site preparation, many of our sites are surrounded by a seed source of invasives, so recolonization is a constant threat. For us, the site goal is another important factor that determines what mix we use. Most of the Meadoway is located in very high traffic residential or commercial areas, where we tend to use high forb seed mixes to make it more visually appealing for the public. A few of the areas we have are more industrial, so we're able to use more grass-based mixes there to help attract different types of wildlife. Lastly, seed availability has been a major influence on what species we're using in our mixes, especially due to the size of the areas we're seeding in a given year. So some years we're buying seed for 25 hectares for this one particular project, which is a massive drain on our very few seed suppliers' inventory levels. Frequently, our orders have multiple species substitutions and omissions due to availability. So what we ideally want isn't necessarily what we get every time. Generally, we try to use species that have historically been found in our project area or at our closest reference sites. As we try to boost diversity, though, and species success, we have incorporated some species that are found slightly south of us, whose ranges are moving northward due to climate change. These species have been proven to successfully grow in our area and are fairly competitive, so they're good at resisting those invasive species we're trying to combat. Our seed suppliers grow and augment seed that was sourced from areas within 1 or 2 seed zones from our site, but that helps ensure that it is locally adapted to our site conditions. Next slide, please. So as the project has matured, we've recently felt comfortable enough to harvest seed from some of our older sections of our restored meadows. Although we acknowledge that these plants maybe don't have the same local wild genetics, we know that they're well adapted to site conditions because they're growing in numbers that warrant harvesting. Because Toronto is so urban and the effects of urban sprawl are so substantial, there's very few wild populations left in our seed zone, so exclusively wild sourcing would be pretty detrimental to those few wild habitats. In the last 2 years, our seed collection has mainly been hand collection of some of our target species within our sites, but we are looking to scale up this year with more mechanical methods. We generally use this seed to supplement our bought seed, especially if our growers are short on some species, and overseed small areas that need a boost. Next slide, please. So our vegetation monitoring team has collected baseline data and yearly post-restoration data from monitoring plots throughout the Meadoway, and then compiles yearly reports demonstrating their findings. Overall, there's been really positive feedback with the increase in species richness and percent cover of these planted species. Obviously, this varies year to year, as you can see on the graph in the bottom, and across the length of the project as different areas are maturing and establishing at different times. We use this data as a tool to drive future invasive species management priorities and to help guide decision-making about species successes within our seed mixes. Next slide. So as we have expanded and adjusted our seed mixes, we've had difficulty balancing the desire between species diversity with the knowledge that we have a much smaller core group of species that we know will actually grow, mature, and outcompete the non-natives. So for example, some mixes contain 28 species, but there's really a core group of about 10 species that we know actually establish as well. Although our goal is to have high diversity of species, we know that they don't all grow and establish in the same way, and some are more aggressive than others. For this reason, we're trying to constantly create new trial mixes that include species at varying percentages and quantities to try to achieve both that species richness and abundance. We know that obviously diversity increases the resilience of ecosystems, but we also acknowledge that if a species doesn't thrive in the space that we're creating for it or gets outcompeted, that it might not be worth investing more time and money into it. So tied to that is the issue of invasive species, as one of our main challenges with urban restoration is the high presence and diversity of invasive and non-native species that end up competing with native planted species for resources. Invasive species pressures have forced us to change and adapt our species that we're using because they can easily outcompete some of the native plants that aren't typically aggressive and fast-growing. We're also working on finding the best early successional native species to hold space in those newly restored areas for later successional species, so that invasives don't have the opportunity to gain a foothold in that space, kind of buy some time for the other species. Finally, with the limited amount of seed vendors within or close to our seed zone, it's been very difficult to get enough local seed to fill the requirements of this large-scale project. Better planning and working with seed vendors to scale up on required species before the project happens would be beneficial, but obviously this is very difficult to do without secure long-term funding on both sides. As our restoration sites mature, we're hoping to turn to harvesting more of our own seed to help mitigate issues with species shortages. Next slide. And that's just a little snippet of the Meadoway project. Thank you for listening, and feel free to reach out if you have any questions.
Thank you for that awesome presentation, Sarah. I don't think we have time for questions right now, but there are a few questions that we'll be pasting into Whova. So up next, Rory Schiafo, who is a PhD candidate at Northwestern University, will be talking about two parts of a whole, sown and non-sown species having different responses to environmental gradients in the woodland restoration of Cook County, Illinois. There we go.
All right, thank you so much for having me here today. I'm so excited to talk to you about my research studying the restorations in the oak woodlands of Cook County, Illinois. Next slide, please. So oak ecosystems are transitional ecosystems that exist between open prairies and closed canopy forests. Next slide. The plant diversity and composition in pre-degraded oak ecosystems was driven by naturally heterogeneous gradients of soil nutrients, canopy openness, and fire regimes. Next slide. However, factors such as fire suppression, invasive species, and land use practices have caused significant habitat degradation in oak ecosystems. Next slide. So to restore these systems, it often requires removing invading trees and shrubs, as well as reintroducing processes such as fire. Next slide. However, to fully reach targets of plant diversity and ecosystem functioning in oak woodlands, the reintroduction of plant species through native seed addition is often required. Next slide. And these communities that we restore through native seed addition differ from their remnant or pre-degraded ecosystems in that they're composed of both sown species, which are the species that we add during restoration, as well as non-sown species, which are the species that persist in a community that occur via natural dispersal and seed banks. Next slide. And so when considering how an environmental gradient is going to impact a restored community, it's important to consider both the sown and the non-sown species in a community. And this is because while these two groups of species may have the same response to an environmental gradient, it's also possible that the sown or non-sown species may have a stronger response, or even that these two groups have contrasting responses to each other. Next slide, please. And so the net effect that an environmental gradient can have on a community, considering all the species together, may range from strongly directional to relatively neutral. And so when we are trying to understand how various site level conditions or management practices are influencing our restoration outcomes, it's important to consider both the sown species and the non-sown species separately. Next slide. So in my work, I seek to investigate how plant communities restored through native seed addition respond through gradients of soil, light, and fire. And I do this through 3 questions that I'm going to walk you through today. Next slide. But first, I'll introduce you to my study system, which is 7 restoration sites in the North Branch of the Forest Preserves of Cook County, which is the county where Chicago is located. Next slide, please. At each of these 7 restoration sites, I established 3 50-meter transects. Next slide. And at each of these transects, I conducted plant community surveys in 10-meter squared plots for a total of 210 plots across all of the 7 restoration sites. Next slide. And I also surveyed the environmental gradients of soil nutrient availability, canopy openness, and burn frequency at each of these transects and plots. Next slide, please. And so for my first question, I wanted to address whether seeded communities have the same response to environmental gradients as communities that are being restored without seed addition. Next slide. And so 13 of the transects that I surveyed are seeded transects. So they are composed of both sown species and non-sown species, whereas 8 of the transects I surveyed are being restored without native seed addition, so therefore they are composed of all species that have naturally dispersed to the site. Next slide. And so what we're going to be looking at here first is the relationship between species richness and these three environmental gradients. In seeded plots will be in purple and unseeded plots will be in orange. Next slide, please. And what we find is pretty interesting. We find that sometimes, as is the case with the soil nutrient gradient, seeded and unseeded plots have the same response to an environmental gradient. However, we also find evidence that seeded plots can have a weaker response to an environmental gradient, as we see with the canopy openness gradient, or that seeded and unseeded plots can actually have contrasting relationships to each other, as we see across the burn rate gradient. Next slide, please. And therefore, when we consider when considering all the plants, all the communities across this restoration landscape, the overall community response to gradients can vary from strongly directional to neutral. Next slide, please. And so next, I wanted to look within communities to understand whether sown and non-sown components of the same community have differing responses to environmental gradients. Next slide. And so for this, I looked at just the 13 transects that are receiving seed addition. And so again, those are composed of sown species, which are any species that are on the seed mix list for these woodlands, whereas non-sown species are the species in these communities that are not on these seed mix lists. Next slide. And what we find is that when looking at just the sown species in these seeded communities, we don't find any relationship between sown species abundances with any of the three environmental gradients. Next slide. However, when we look at the non-sown species in these same exact communities, we find that non-sown species increase in abundance across a canopy openness gradient. Next slide. And decrease in abundance across a burn rate gradient. Next slide. And so overall, we're seeing this stronger response to the gradients of canopy openness and burn rate among the species that are not sown into restorations. Next slide. And so I wanted to try to understand this a little bit more. So I ask whether species, the same species, have the same response to environmental gradients when they're sown compared to when they occur via natural dispersal. Next slide. So to do this, I looked at a select group of 28 species from the seed mix list that's used to restore these woodlands that occur in both seeded and unseeded plots. So when these species occur in seeded plots, it can be assumed that they're there because they were sown into a restoration. But when they occur in unseeded plots, it can be assumed that they're there because they've occurred there naturally through dispersal or seed banks. Next slide. And what we find is that when looking at these 28 species in just the seeded plots, so when they occur there through restoration actions, species abundances do not vary across any of these environmental gradients. Next slide. However, when looking at these same 28 species in the unseeded plots, so when these species occur in a community through natural dispersal, we see that species abundances do increase across environmental gradients. So this gives us some idea that these species are responsive to these environmental gradients. However, next slide. Again, we're seeing this stronger response to environmental gradients when a species occurs via natural dispersal and not restoration. Next slide. So just to summarize, do seeded and unseeded communities have the same response to environmental gradients? Well, it depends. Sometimes they do have the same response, but we also find evidence that unseeded communities can have a stronger response to a gradient, and that sometimes seeded and unseeded communities actually have contrasting responses. Next slide. And thus, the overall kind of net effect that an environmental gradient can have on all the communities on a restoration landscape can range from directional to neutral, depending on these different relationships. Next slide. Then when looking within seeded communities, we find that sometimes the sown and the non-sown components of a community can have the same response, but we also find compelling evidence that non-sown species often have stronger responses to the environment compared to their sown counterparts. Next slide. And finally, when we look at individual species and we try to understand their responses to environmental gradients when they're sown compared to when they occur via natural dispersal, we find that across all three of the gradients that we looked at, species have a stronger response to the environment when they occur via natural dispersal, as opposed to when they're sown into the restorations. Next slide, please. So what does this mean for the restorations of oak ecosystems and restorations more broadly? Well, one, it suggests that the differing responses to environmental gradients between non-sown and sown communities and species should be considered and can help us understand restoration outcomes. It also suggests that the non-sown species are likely to have a strong response to changes in environmental conditions, for better or for worse. And finally, sowing species into woodland restorations may offer the possibility to overcome constraints in species establishments and success that may be set by environmental conditions such as soil properties, light availability, and burn intervals. Next slide, please. And with that, I just want to thank my graduate program, my funders, the Forest Preserves of Cook County, of course, my many research assistants, and my committee. Thank you very much, and please reach out with any questions.
Thank you so much, Rory. I think we have time for one question. Is species richness in the plots only for native species, sown and non-sown?
So it's both native and non-native. And so something we're looking to do in further analysis is look at the compositional differences between the sown and non-sown species, kind of with the idea that most likely the non-sown species in the communities are more likely to be non-natives or ruderal species, which would explain things like the decrease in abundance with an increase in burn frequency. Yeah.
Thank you.
Thanks.
So there are more questions. They'll be moved over to Huva. We need to move on to our next speaker, who is Marissa Lane-Massee, a graduate student at the University of Oregon, who will be speaking about an agroecological restoration case study on Oregon hazelnut orchards. So you can go ahead and turn your video on.
Can you see me?
Yes.
Okay, awesome. I will be presenting today about using seed-based restoration to improve orchard systems and utilize land sharing in Oregon hazelnut orchards. Next slide, please. So my name is Marissa Lane-Massee. I'm a graduate student at the University of Oregon in the Hallett Lab. I am funded by Western States and the USDA, and I have a COI statement that I need to share with you. All research from this presentation has been conducted on Lane-Massee farms. That is my family farm. All right, next slide. Okay, so a little ecological background for everyone. In Willamette Valley ecosystems in Oregon, there's not much remaining like native habitat intact. And there's a large push for oak ecosystem restoration. And there's this weird phenomena between hazelnut agriculture and oak conservation. They share pests and they share spaces, and it's an ongoing fight against agriculture and against conservation from both sides. So my main work is to see how we can meet in the middle with the resources that we have today. Is there a way that we can meet the needs of the ecosystem and meet the needs of the farmers? And so land sharing in agricultural systems has typically been difficult. Perennial cropping systems seem to have the best chance for sharing viable space, and orchard systems may offer this heightened chance for success. Maybe orchards can get some functionality from restoration ecology methods. Next slide, please. So in hazelnut orchards, or just orchards across the Western US, bare soil management practices are used, and there's a number of ways to get to them that include herbicide spraying, ground scraping, mowing, stuff like that. And then in Oregon, where hazelnuts are the primary nut crop, these practices are also used. Next slide, please. So this makes a good case study for orchard systems being the perfect place for land sharing. These orchards are already void of vegetation. They may only need bare ground once a year. And then many orchards would also benefit from the ecosystems provided. Like in California, almonds could really use some additional pollinator habitat. All orchards could use soil health benefits. And there's this shared space for native diversity that is actively managed without additional input from farmers. Next slide, please. So part of the work that I've been doing, I did a small pilot project in 2019 through 2021, just to test whether these cover crops even work. You know, we compared conventional cover crops to conservation cover crops. You know, do they have differing effects in hazelnut orchards? Can they be worked with? You know, just what's the feasibility of this? We also looked at like disturbance resistance, pollinator forage, and then soil moisture on top of that. And then we really came to the conclusion that the conservation cover was the way to go. We expanded a pilot project from 2022 to 2023, just to refine the conservation cover, and then improve management strategies, and then look at the phenology, and then really combine this to create a robust native seed mix. And then now we're working on a multi-orchard project, testing multiple seed mixes across multiple orchard canopy densities. And then we're looking at the economic factors behind that. Next slide, please. So why did we go with conservation cover? We pretty much found that the native plant phenology matches the management cycles of hazelnut agriculture a lot better than the conventional cover cropping phenology. And this match phenology is perfect for harvest in hazelnut orchards. You can basically mow down your cover crop right before harvest. The nuts can fall on the ground. And then during a dry harvest year, it's basically bare ground against bare ground, seed mix versus control. In a wet year, you do have some vegetation regrowth in the seed mix, but it really actually facilitates crop pickup in the wet years. Next slide. So we also found that the conservation cover really outcompetes weeds. It can outcompete weeds for up to about 85% plot cover dominance. And this conservation cover can grow in heavily shaded areas where conventional covers can't. And then they have all the benefits of conventional cover crops plus more. So you can see in that graph on the right-hand side, in the blue is our target cover, that's the seed mix, and then in the green or in the purple is the weed cover. And you can see that's really stabilized by the target cover. Next slide, please. And then populations of conservation cover can also handle intense amounts of disturbance. You know, they've adapted to these ecosystems over time, and conventional cover crops just can't handle the amount of disturbance that the conservation cover can. And so in our pilot project, we studied this against an unmanaged plot, a flailed plot, and a scrape plot. Flailing is just mowing. And we found that the native perennial forbs can really, really handle intense amounts of disturbance. The native annual forbs do their thing, as you guys know, and the conventional cover crops really couldn't handle much. Next slide, please. So the way we figured out our conservation cover seed mix, we looked at orchard survival, like what species will survive in the given orchard system that we're working in. So can they survive the sun to shade ratio? Are they easily dispersed by machinery? And are they competitive towards weeds when in a semi-invaded community? Next slide, please. So we came up with this general species pool filtering method. We looked at, you know, what can you seed into bare orchard floors? And then we went through a bunch of plant filters and agricultural filters, looking at light availability, the different mechanical treatments, and then, you know, do they really prevent erosion? Can they increase soil moisture or stabilize it? Can they outcompete with weeds? And, you know, the ever looming question, what about harvest? And then, you know, we narrowed our cover crop species pool down and then came up with a list of compatible cover crops. Next slide, please. So we have our orchard survival. Next slide, please. And then we needed to figure out, what's our management timeline? When do certain management activities take place? And can we select species that can handle disturbance but are dormant or in a vegetative stage for especially important activities like harvest or required spraying? Next slide, please. So we had to come up with this time frame for orchard management and cover vegetation. And so we just mapped out the hazelnut phenology. We looked at harvest and then the different mechanical treatments that are needed. We looked at broadcast pesticide applications, and then we saw, when can you put cover crops in? When do they need to be terminated by? And what are our management strategies that we can exclude or include in our treatments if we go further with this project? Next slide, please. And so we figured that out, and then we started working. And we pretty much found out that standard restoration ground prep is the way to go. And many landowners already have pre-prep soil, so you may not need to do a lot of work. And then there's not any need for any additional equipment. Farmers may already have access to seed drills. And then if not, a belly spreader works just fine for multiple acres. The hardest part we found was the seed selection, seed sourcing, and then syncing that orchard management and plant phenology. Next slide, please. So if we go and we want to look at broader impacts, the conservation cover possibility could improve orchard systems to help farms economically and ecologically. Economically being, it could really prevent a lot of labor costs. It could stop a lot of the herbicide spraying, a lot of the mowing. And then ecologically, we did a pollinator study. I didn't include the data here, but we basically figured out that there's like 30 different native pollinator species that can be drawn to the cover crops, depending on what species you select, versus the conventional cover crops, which bring in maybe 4 different pollinator species, native pollinator species. And then, these can really improve conditions for the surrounding ecosystems. And then if adapted across many different orchard types, it could vastly increase the native flora and fauna habitat across the Western US. So on the right, you can see the tree nut production in the Western US that has similar management practices. So in Oregon, there's about 90,000 acres of hazelnuts. That's about like 8% of the Willamette Valley. And then in California, there's about 2.23 million acres of walnuts, almonds, pistachios, and pecans. I don't know how much of California that is. But these seeds, if grown on the farm, could be sold to dramatically increase the amount of available seed for restoration practitioners. And this could greatly help the restoration community as a whole. Next slide. So thank you for coming to my presentation. Questions?
Thank you so much, Marissa. That was an awesome presentation. I don't think we have time for questions at this time, but there are quite a few questions that will be transferred to Huva.